Biotechnology and Food Science Congress. I am Dr. Emenuga Veronica a lecturer with the Department of Medical Laboratory Sciences, University of Nigeria Enugu Campus, Nigeria. The title of my work is Assessment of Health Risks Involved in the Consumption of Aflatoxins from Peanuts and Cashew nuts are commonly consumed nuts due to their high nutritive values and delicious tastes. Sometimes these nuts are contaminated with fungal organisms which produce toxic substances like aflatoxins. Aflatoxins are mycotoxins produced by secondary metabolism of some filamentous fungi. There are three main genera of fungi that produce mycotoxins. They are Aspergillus, Fusarium, and Penicillium. There are four types of aflatoxins: aflatoxin B1, B2, G1, and G2. Aflatoxin B1 is the major potent toxin. Aflatoxin B1 are mainly produced by Aspergillus flavus, Aspergillus nummus, and Aspergillus parasiticus. Where can we find these aflatoxins? They can be seen in seeds that are rich in protein, fats, minerals, low water content. They can be seen in dry fruits, in nuts, and in grains such as maize, millet, wheat, rice, groundnuts, and cashew nuts. There can be fungal contamination when these seeds are still on the tree or in the root, as in groundnut that is a root nut. There can be an unhygienic handling of these nuts during harvesting and during processing. In that method, these nuts can be contaminated. When there is poor packaging of these nuts in poly bags and exposure of these nuts to the heat of the sun during hawking, of course, there will always be a recondensation of moisture from these poly bags which are used in packaging these nuts. When there's this recondensation of moisture on the surface of these nuts, it of course enhances the growth of molds, and this will create food safety problem. Justification. Aflatoxin B1 is very toxic. It can be converted to aflatoxin 8,9 epozoites in the liver, and it can cause DNA degeneration. By so doing, it can also lead to spontaneous abortion in human. It can cause stunted growth in plants. It can cause aflatoxicosis in livestock and domestic animals. Aflatoxin B1 is so potent that it can cause acute or chronic diseases like hepatocarcinoma. It can cause cirrhosis of the liver. It can cause mutagenic and teratogenic cancers. Aflatoxins are heat resistant in nature. Therefore, most of these nuts that are being roasted can still have aflatoxins in them, because roasting most often might not be able to eliminate these aflatoxins. Hence, the aim of this work, to assess the health risks of aflatoxin consumption from nuts. Materials and method. 50 samples of packaged peanuts and 50 packaged cashew nuts were bought from 5 different markets in Enugu metropolis between the months of June and July 2019. These different samples were sorted out, and the infected seeds were seeded or inoculated onto sabroid dextrose agar, which contains 0.01 chloramphenicol. These plates were incubated at 25 degrees centigrade for 7 days. When there was fungal growth after 3 to 4 days, the identification of fungal isolates started with macroscopic and microscopic means. By macroscopic and microscopic characteristics, the fungal organisms were identified. Other identification tests were also done using standard procedures. There was determination of the percentage fungal occurrence. This was done according to the method described by Atanda et al. 2002. The moisture content of the nuts were determined according to the method of Wu et al. 2013. The detection of the total aflatoxin that is present in the different nuts, it was done by the use of ELISA method using the protocols that was described by Adetunji et al. 2014. The level of exposure of the consumers to aflatoxins were also determined as described by Adetunji et al. 2014. Estimation of liver cancer risks was done using albino Wistar rats. 20 albino rats that were laboratory reared, weighing 280 to 340 milligrams, were housed in 5 cages of 4 rats per cage in the laboratory. All the rats were screened for hepatitis B viral antigen and alkaline phosphatase test before they were being fed with these nuts. They were fed with these nuts according to different concentration designated on their cages for 30 days. After the 30th day, these animals were also bled again, and hepatitis B viral antigen and alkaline phosphatase test were also done again, so as to use it to compare the initial one that was done at the beginning of the test, of the experiment. Histological study of the liver tissues of the rats. One of the animals from each cage were sacrificed at the onset of the experiment, and their liver tissues were studied as a baseline study. This can be seen in figure 1, or in the figure 1 of this work. After 30 days of consumption of the nuts, the animals were sacrificed and histological studies was done on their liver tissues. This was also shown in figure 2. It can still be seen in the slide. Statistical analysis that was used for this study was SPSS version 25 software, and the data that was obtained from this work was subjected to analysis of variance at 95% confidence level to test for the significance of our results. Results. The mean percentage moisture content of the nuts, that is of the peanut and the cashew nuts, were recording 5.26% and 3.7% respectively. When they were tested, it was found not to be statistically significant. 6 fungal isolates that were isolated from these works. They are Aspergillus parasiticus, Aspergillus niger, Aspergillus flavus, Penicillium species, Fusarium species, and Rhizopus species. Aspergillus flavus and Aspergillus parasiticus were high in their occurrence, having 45 and 36% occurrence respectively. This is shown in table 1. From this table 1, it could be seen that Aspergillus flavus had 45% occurrence, while Aspergillus parasiticus had 36% occurrence. The lowest fungi that occurred at the period of this experiment was Rhizopus, which had 1% occurrence rate. Table 2 shows the aflatoxin concentration of the nuts, which was performed using ELISA method described by Adetunji et al. 2014. From this table, we can see that the aflatoxin concentration on peanuts ranged between 26.0 to 32.48 microgram per kilogram. while that of aflatoxin concentration in cashew nut ranged between 4.6 to 10.9 microgram per kilogram. Of course, the aflatoxin concentration in peanut is far much higher than the recommended aflatoxin concentration, which should be 20 microgram per kilogram. The assessment of exposure level of the consumers to aflatoxins was done using the method of Adetunji et al. 2014, and it is done by multiplying the quantity of aflatoxin in the nut with the average consumption rate and dividing the value that's gotten with the assumed body weight of a Nigerian adult. And the assumed body weight of a Nigerian adult, according to WHO 2008, is 60 kilograms. The level of exposure of these consumers to aflatoxins when it was done is shown in table 3, and it could be seen that the range of level of exposure of consumers to peanuts is between 25.35 to 28.15 nanogram per kilogram body weight per day, while that of the cashew nut ranged between 0.28 to 38 nanogram per kilogram body weight per day. Biochemical and histological studies of the rat tissue that was done. The biochemical tests of the rats that were done showed that the hepatitis B viral antigens were all negative even at the onset of the test, and also at the end of the experimental period, that is at the end of the feeding period, which was 30 days, the result that was also gotten from the hepatitis B viral antigen test was also negative. The alkaline phosphatase test that was done on the animals shows that there was a little increase in those rats that were fed with aflatoxins from groundnut. Those animals that were fed with cashew nuts did not have any difference in their alkaline phosphatase test results. The liver tissue of the rat fed with groundnut revealed granular appearance of hepatocytes and severe hydrophilic vacuolation degeneration. With this, you can say that there was a decrease, a degeneration of the liver vacuoles, while there was also granulation of the hepatocytes of the liver tissues. This is shown in figure 2. That is, these are the effects of the aflatoxins on the liver tissue. Figure 1 shows the normal liver tissue from the rats that were not fed with any of these nuts, while figure 2, the H here shows the granulation of the hepatocytes, while the V here shows the degeneration of the vacuoles. That is from the liver tissues of rats that were fed with aflatoxins from the groundnut. Discussion. The moisture content of the roasted peanut and cashew nuts were above the permissible recommended moisture limit of 5%, according to WHO. While in this work, we were getting a moisture content that was ranging between 22 to 32.48 nanogram per kilogram. It could be seen that the moisture content of the peanuts in this work was actually higher than the recommended moisture content. This could be as a result of the poor harvesting or improper processing method. It could also be as a result of the use of inadequate materials for packaging these nuts. Nuts tend to absorb moisture from the surrounding atmosphere. This was reported in the work of Oladapo et al. 2016. So when you look at the moisture content value that was seen in this work, it must have been as a result of absorption of moisture content from the surrounding or from the packaged cellophane bag that was used in packaging these nuts. All the peanut samples in this work had aflatoxin concentration ranging between 26.0 32.48 microgram per kg, which is actually higher than the recommended permissible limit by the Standard Organization of Nigeria. The standard limit should be 20 microgram per kg for unprocessed peanut. Low concentration of aflatoxins from cashew nuts ranging between 4.6 to 10.9 microgram per kg was reported in the cashew nuts that we have in this study. But this does not mean that it is the normal recommended concentration. It is still above the recommended permissible limit. Risk of exposure of aflatoxins in peanut consumption was actually high, and it was ranging between 22.53 to 28.15 nanogram per kilogram body weight per day. This report, although it collaborates with the work of Oyedele 2017, who also reported a PDI value of 91.2 nanogram per kilogram body weight for groundnut consumers in some zones in Nigeria, it is still all above the permissible limit of groundnut consumption. The risk of exposure of cashew nut consumption was also high. It also ranged between 0.16 to 0.38 nanogram per kilogram body weight. All this is still above the required standard value. The required standard value for cashew nut consumption, that is the required standard exposure risks for cashew nut consumption, should be 0.017 nanogram per kilogram body weight. But what we have in this work was actually higher than this value. Conclusion: the study showed that nuts were contaminated with aflatoxigenic fungi, which poses health risks to their consumers. The low value that we got in the cashew nut was still above the permissible limits. But even at low values, they are still very, very hazardous to health because smallest molecule of aflatoxin B can evoke changes in human DNA, leading to mutation and cellular proliferation. Aflatoxin B has not been recorded to have any safe dose at any value above 0. In the animal pathogenicity tests done, the slight degenerations of the liver tissues of the rats fed with groundnuts revealed that the aflatoxins have adverse effects on health of both human and animal. Recommendation: it is therefore recommended that adequate sanitary measures should be encouraged in the handling and storage and packaging of foods and nuts, and these measures should be enforced and regularly monitored so that people will not deviate from it in order to reduce the menace of food-borne diseases and mycotoxicosis among the populace. The slide below shows some of the fungal organisms that were isolated in this work. We can see the Aspergillus and Parasiticus, Fusarium species, the Penicillium, the Aspergillus niger, and the Aspergillus flavus. All these are some of the fungal organisms that were isolated during the course of this work. Thank you for listening.